Thank you very
Thanks,
much.
Ari.
Yeah, it's a bit of a mouthful. I apologize about that. But yeah, so it's quite convenient that I'm following on from Daniel. We didn't plan this, but I think the talks go quite nicely together. So yeah, I'm going to be talking about a platform that we've developed utilizing causality inferencing and large language models to help kind of streamline some of the candidate gene target lists that we generate and generate some actionable next steps for us to take into the lab. Do that one. So apologies, I'm going to sound a bit like a broken record here because it's very similar to Daniel's slide. But yeah, just a quick primer. So at Lonza, we're concerned with the manufacturing of our customers' molecules. Again, it's done in Chinese hamster ovary cells, CHO cells, and using large-scale bioreactors. Again, the kind of production pipeline looks a bit like this. We'll start with the genetic construct and cell line generation. We'll then move through to scaling the culture up, and then finally into purification, formulation, and distribution back to the patients. Again, in the research group, we're interested in engineering the CHO cells to be able to alleviate bottlenecks and improve the pipeline at various different places. So, for example, we might want faster growing clones, or we might want clones that produce high quality therapeutics. And so we can do that by targeting the original cell line. But in order to, I apologize, it's covered up. But in order to do that, we of course utilize omics. But we need to go a bit further than that and actually generate some real biological insight and mechanistic understanding of what the candidate genes are actually doing, the roles that they're playing, so that we can have some more confidence and filter down the lists of genes that we have back then into the lab. So we are often presented with situations like this. So we might, again, take two cell lines, both producing the same molecule. One's performing very well, one is not performing very well. We'll extract some material, might send it for proteomics or transcriptomics. We'll do some differential expression, and we'll end up with something like this. So hopefully people are familiar with this type of plot, volcano plots. Essentially, every highlighted point, every colored point is a statistically significant gene. The red ones are low titer markers, and the blue ones are high titer. Titer just means the amount of product produced per cell. Basically, we have thousands of candidate genes here. We don't know, we can't just take this into the lab and systematically knock out every single one of them because we'll be there for years. So we need ways to basically take these lists and generate kind of concrete hypotheses, concrete next steps, so that we can have higher confidence and shorten the timelines that it takes to go from new molecule and a new cell line to engineered. So ultimately, the point is that we often stop at this stage, or at least get to this stage, and these gene lists show us what changed, but they don't show us why, and that why is what we need to be able to translate back into the lab. And so to address this, of course, there are existing methods, namely causality inferencing. So it's all to do with cause and effect. So with forward causal inferencing, you induce a cause and measure the effect. So again, we might be taking some of those genes from that volcano plot, knocking them out and seeing how it changes the cells. With reverse causal inferencing, we then go the other way around. So we start off with the effect and work our way back to try and deduce the cause. And so, again, taking that same example, we might get a high versus low product expressing cell line, generate some candidate genes, and then this is our measured effect. So we're trying to then work back to the cause. This might look familiar. I'm sure people are used to gene set enrichment analysis, that type of thing. And it's basically where we can take that effect, take those measured genes, and measure them against other signature sets. So in this case, we can take public signature gene sets that have been curated by the community. So it might be pathways, or it might be just standard literature, gene expression omnibus or expressions atlas, anything like that. All of these signature sets will then have the associated metadata for publications. They'll have a manuscript, or for curated pathways, they'll have gene ontology analysis. And this gives us the why. This gives us that mechanistic and biological insight. But it's a bit of a catch-22 because, just as we can end up with thousands of candidate genes, we can end up with thousands of overlapping pathways or thousands of overlapping experiments. And so this is where we can introduce the large language models. Won't spend too long describing large language models. I think everyone should be probably familiar with them now. But we can basically take all of that metadata that comes from these overlapping experiments, pass them through large language models, which just large text synthesizers, and give us a single kind of refined report with actionable, concrete next steps. As well, it can they can uncover signals that might be missed by human curation. But I mean, both of these things have existed by themselves, so why do we actually need to put them together? It's kind of a quite frequently asked question. LLMs have a tendency to hallucinate sometimes. They will tell us what they think they want us to know. If we ask them to find us biomarkers, they'll find a way to find us biomarkers, even if they're not of any use. So pairing it with the statistical rigor of the causality inferencing gives it that grounding in truth, so we can always guardrail it. And again, I alluded to this earlier, but when we're running RCI, we might end up with hundreds of or even thousands of overlapping experiments. And this TISNI you can see here, again, apologies, it's quite hard to see, but each of the points on here is basically a world map of one of these public signature databases, Expressions Atlas in this case. All of the points is one experiment. There's around 8,000. After, again, taking that same example of the high versus low expressing cell line, getting the DGEs and running the RCI, each of the highlighted points here is a statistically significant experiment. And so there's around 170 here. So there's 170 manuscripts to read. It would take me a very long time to do that, several months probably. And so by using the large language models, we can slash that time quite dramatically by getting, offloading it to GPUs instead. And so with this kind of methodology, we get the best of both worlds, basically. So we get, it's modality agnostic. So basically anything that we can run against the knowledge, using the RCI. For example, genes, we can get candidate genes from multiple different types of omics, as Daniel just described. So it could be transcriptomics, could be proteomics. We end up with a list of genes to work with. It makes this effect to cause easy. We just start single pipeline. We start off with candidate genes, and we get out at the other end a single tailored report that summarizes all of the signals and tells us what to do in the lab, or what we can look at in the lab. And of course, then it reduces the time from days down to minutes. So I'll now talk a bit about our actual implementation of this pipeline. And so we built graph-powered RCI. So we basically, again, start with the initial gene list. Of course, everything will have, each gene will have a state change based on its log2 fold change or whatever it might be. So we'll know if it's going up or down. And of course, it will have a p-value attribute from the prior analysis. We first, of course, select the foreground and the background genes. And then using those foreground genes, we screen it against our graph database. In here, we've got two different types of nodes. We have upstream and downstream nodes. So the downstream nodes represent the genes, and the upstream nodes are collections of genes or the experiments. And so it looks something like this over here. So the dark blue blobs would be the experiments, and the upstream nodes, and the light blue ones would be the genes. And you see that some genes can be shared between experiments, and then the edges would correspond to their directionality in that respective experiment. And so we can use those query genes to extract the overall experiments and then run the enrichment tests. And so for each input gene list, we'll get a set of results. Each experiment, overlapping experiment, will have two metrics. We have concordance and richness. So concordance is based on the state change agreement. If all of the genes in the input gene list are going up, and they're all also going up in the respective experiments, then it has a lower concordance value. And the richness is then just your Fisher's overlap. So the traditional gene in set A and set B. And then, of course, we have all of the associated metadata along with that. And so right now we kind of have two layers, you could say, in the knowledge base, and they're mirrors of some public databases. So we have Expressions Atlas, which is a collection similar to Gene Expression Omnibus. So it has for example, experiments like treatment of cell line X with chemical Y, and then it has a signature set, so you know exactly what the measured effect of that is. And then we also have MSigDB, which is the molecular signatures database. So this is more curated pathways, things like transcription factor sets. And so what we do is we run the RCI against both of those layers. So we get two sets of results, one for Expressions Atlas and one for MSig. And so they'll have the multiple experiments and multiple manuscripts or gene ontology analysis. We then supplement those two kind of data sets with the original input gene list. So we give it the original measured effects along with the gene state changes. We also provide a bit of the study background. So again, in the case of the high versus low producing cell line, we would give it a small kind of abstract describing what we did in the lab and how the material was generated, so that the large language model can better contextualize. We then pass that kind of data package through a first layer of LLM or a first agent. And this agent gives us two independent intermediate reports. So we get one tailored for the Expressions Atlas analysis and one for the MSig analysis. And the reason that we do this is that we found that it retains detail much better than trying to just stuff everything into the same context. A bit like humans, I guess, the higher the information entropy, the more loss, information loss. So we separate them out through a first layer. We then, of course, take that intermediate layer and those two reports and pass it through a second stage of summarization. And this is what gives us our single refined report. And here is where we ask it to provide next steps in the lab and help us to design follow-up experiments based on that the original study. And so each of the agents will have independent instructions. But at every level, we always ask it to provide the experiment reference. So again, we don't want the LLM to go off on tangents and start drawing on its training data. So we ask it to always reference studies from the original input data sets. So it provides the accession numbers so that we can follow up and verify things if need be. But we also ask it to highlight contradictory evidence. Again, I mentioned earlier that LLMs just generally like to please us. So by giving it this kind of grounding, we get a balanced response as well. So it also will tell us things that don't make sense according to the data. And so we wanted to put this to the test to see how well it worked. So we used a public data set. This is it was an experiment. Some material taken from a producing CHO cell line versus a non-producer, standard IgG1 monoclonal antibody. It was temporal, so there were 3 different days that the material was taken on. So we had 3 different sets of differentially expressed genes, day 1, 3, and 5. And so we ran into, I should also mention that the data, this particular study was published after the LLM training window. And it's also not in any of the knowledge bases, EA or MSig. So we were sure that there was no cross-contamination. But we took those 3 differentially expressed gene lists and ran them through then the RCI LLM curated pipeline. And then, of course, we'll end up with 3 independent summarized reports from those 3 different days. So we have then a third layer that works on multiple treatment or temporal time point summaries. So we're able to do longitudinal data sets as well. And the whole thing is built with Nextflow for scalability and reproducibility as well. So I'll now talk a bit about the evaluation and what we actually get out at the other end. So this is just a small snippet of the final report. You can see that the LLM draws on the statistics from the original RCI run. It gives us the accession numbers of experiments that it's drawing conclusions from. It gives us specific gene lists to work with and specific pathways that are enriched in the RCI results. And of course, it then also points out some of the contradictory evidence as well. So it gives us grounding against what we would expect to see. It's quite hard to quantitatively evaluate this. We tried some cosine similarity things, but we're comparing one result set against many, so it doesn't work too well. So what we did is ended up using an independent expert, which was basically a deep research model. And we gave it the original manuscript that we took the data set from, and then the report generated by our platform, and asked it to basically provide an evaluation. So you can see that the platform is kind of generally highlighting some real biological signals, and also things that the original manuscript didn't pursue, not necessarily because they didn't notice them or whatever, but nonetheless, identifying signals that weren't originally there. It was also exploring things that are kind of mechanistically credible. So some of the genes and pathways that it was telling us to follow up with and to target in the lab, are credible for boosting yield in this case. But of course, the ultimate litmus test will be to take it into the lab and actually try out some of the genes and try out some of the steps that it was giving us. So that will be the up and coming, maybe for the next conference. And so just to finish off, we have this Nextflow-based RCI LLM pipeline, can convert longitudinal or multi-treatment gene sets into concrete hypotheses and actionable next steps. And the initial evaluation kind of reveals that it's able to provide fresh insights and also draw upon real facts and little hallucination. These slides are a bit old, but we do want to explore some more elaborate ways of providing the grounding from the abstracts. So initially, right now, we were using context stuffing, basically putting everything into the context window. But we'd like to, or we have explored some, but we'd like to go a bit further with vector stores, for example, and see if it makes a difference with agentic RAG, for example. We'd also like to, of course, expand the knowledge base beyond genes, so we can add more downstream nodes or entities into the knowledge base that represent, for example, metabolites. And we can then go from omics data sets with metabolite targets all the way through to the pipeline and ultimate result and summaries at the end as well. Of course, I'm just the messenger, so thanks to everyone who contributed to the project. Some of the names are here today. Yeah, and any questions?
Thank you, Harry. Any questions for Harry? Yeah.
So thank you both, first of all, for two great presentations. So you commented a bit on how hard it is to quantitatively evaluate these LLMs, which I would imagine is a big problem, because also the choice of which LLMs that are so fastly evolving can be hard. So do you have like a more quantitative process for this? Are there like specific public benchmark performances that you look at, or things like this?
Well, generally, I mean, in terms of model selection, we generally want to work with state of the art. So we, of course, pick the best ones that we can. We're currently evaluating kind of reasoning models against non-reasoning. Reasoning models tend to perform a bit better, but only for some of the agents. So some of the layers that I described on the other slide, like they work better than just non-reasoning models. In terms of then the assessment, yeah, it's quite difficult because we're going from like, I mean, in the case of this, we're going from like, one summarized report to potentially thousands of manuscripts. And so it's very difficult to do any, actual quantitative comparisons against that, like cosine similarity. But we did try it, and yeah, it just didn't. It's not very representative. So we fell back onto using more LLMs, basically. So
great, thank you.
Thank you.
Any other questions? All right. I have a question, actually.
Sure.
I'm thinking about the compute capacity that you need to implement some of these. Can you speak to that?
Yeah. So we again, we use kind of batch compute. Of course, we use it with Nextflow, so a lot of it is abstracted away from us, really. It's not an awful lot. I mean, the most expensive would be the inference. But yeah, I think that's pretty much, you know,
Yeah.
sorry, yeah.
Yeah, great. No, thank you so much.
Cool, thank you very much.
Thanks.